Okay, in this video, we're going to look at the structure and analysis of polypeptide systems. These are systems that involve two or more amino acids connected through a peptide bond. Okay, so what's that term peptide bond? You're hearing it for the first time probably. Okay, peptide bond is just the carbon-nitrogen bond connecting one amino acid unit to another amino acid unit, okay, as you see here in this diagram. Okay, a peptide bond is really just an amide bond, the carbon-nitrogen bond of an amide. Okay, this peptide that I have depicted here is a tripeptide. This first peptide amino acid here is a phenylalanine. Okay, let me just turn this over to a printer. So this is a phenylalanine residue. Okay, right here we have a cysteine residue, and right here we have an asparagine or a glutamine residue. Okay. All right, so notice how in the phenylalanine we have a free amine group. This amine doesn't have a C double bond, this nitrogen rather, does not have a C double bond O attached to it. And at the opposite end, we have a free carboxylic acid group. Okay, and this carboxylic acid group, again, does not have a nitrogen attached to this carbon. It's just a free carboxylic acid group. We call this end that has the free amine group, we call it the N-terminal end. Okay, and this end that has a carboxylic acid, we're going to call it the C-terminal end. Okay, one way we can write this protein sequence, and I'm going to be using three-letter abbreviations, is just take the NH3+, then the three-letter abbreviation for the N-terminal amino acid, the middle amino acid, and the C-terminal amino acid, followed by COO-. There's no ambiguity as to what's the N-terminal and C-terminal end when written this way. However, in conventional protein nomenclature, you just write it from left to right, where the left side amino acid is the N-terminal and the right side amino acid is determined to be the C-terminal end. So we write it left to right, N-terminal to C-terminal. Okay? All right, so here's a common type of problem you might see. It'll take you longer to do this, so what you want to do here is pause the video and go ahead and draw this compound. But here I give you the structure of the free amino acids and ask you to draw a pentapeptide that has a sequence leucine, lysine, arginine, aspartic acid, and valine. So leucine has to be your N-terminal amino acid since it's on the left, and valine has to be your C-terminal amino acid since it's the one that's on the far right. Okay, so what does the pentapeptide look like? This is what you should have right here. Okay, again, the leucine here, lysine here, let's go to, lysine here, arginine here, aspartic acid here, and valine as the N-terminal amino acid. Let's look at some common wrong answers that we see in a problem like this, okay? All right, so here's a common wrong answer here. And it arises from the fact that when we have lysine, we have two amino groups, one in the alpha position like any other alpha amino acid, but we have an amino group on the side chain. So what the problem here is that we have used the side chain amino acid, or side chain NH2, to make our peptide bond. And this is not the way proteins form. This is something that can happen, but this would be called an abnormal linkage. The normal linkage is to use only the alpha amino acid as part of the linking system. Okay, another common answer that's not really a good answer at all is that we show something that has oxygen-nitrogen bonds, okay? And this is not a feature of proteins. Remember that when we make an amide, we take an amine and a carboxylic acid, and we make a carbon-nitrogen bond, and we eliminate water, okay? So here, we're not really eliminating water, we're eliminating actually H2, and this is not very favorable. The oxygen-nitrogen bond is really not all that strong, and it's likely that a compound that has multitudes of oxygen-nitrogen bonds like this would be a very unstable structure. Okay, all right, so don't make these mistakes if you see a question like this on an exam. Remember that even if there's two amine groups or two carboxylic acid groups, we always use the alpha amine group or the carboxylic acid that has an alpha amine group as part of our continuing peptide chain. All right. Protein structure determination is a rather complex operation. There are a variety of techniques that you can use. Since this is not really a biochemistry class, I'll just introduce one that makes really good sense mechanistically, and this is called the Edman degradation. So in the Edman degradation, we are going to take a peptide, in this case a tripeptide, and treat it with phenyl isothiocyanate followed by trifluoroacetic acid, and this is going to make a type of compound known as a hydantoin. And it's actually going to give us, the hydantoin arises from the N-terminal amino acid. We're going to basically clip off the whole N-terminal amino acid unit right here. And we're going to be left with a dipeptide composed of everything except the N-terminal amino acid. So since we started with a tripeptide, the Edman degradation is going to give us a dipeptide. Okay, the mechanism here is complex, and this is not something I guess I would really want to ask on an exam, because assigning the partial credit for almost but not quite correct answers would be a real nightmare in a question like this. Okay, so step one, we're going to take the N-terminal amino group and we're going to react it with the phenyl isothiocyanate and make this compound here. This is called a thiourea type compound. And in step two, we're going to isomerize, or we're going to treat this with trifluoroacetic acid, and a series of reactions is going to take place. There are a lot of mechanistic variants that one could write here. This is not necessarily the mechanism, but this is a perfectly reasonable. Okay, so we're going to protonate the N-terminal, and it's going to be attacked by sulfur. You're probably wondering, there's a lot of oxygens in your average protein. Why just that one? Well, in fact, all of them get protonated to some extent, but only this one has a sulfur 5 atoms away, just already set to make this nice stable 5-membered ring here. All right, we're going to lose the proton and make this structure here, and we're going to do a proton transfer from the trifluoroacetic acid. This is going to protonate this alcohol, and I'm going to draw that same compound here. That protonated alcohol that we generated there is now going to cleave, and we're going to, here at this point, we're going to do the all-important separation of the N-terminal amino acid from the rest of the chain. Okay, so that bond right there is cleaved. we're going to get something that's not quite the hydantoin. We're going to get initially this structure right here, which is going to do an isomerization. Okay, so a series of steps. I've just written here that we're going to make the hemiacetal. We're going to open it and reclose it with nitrogen to make the more stable ring system, which is here, which is the hydantoin. Okay, so we're initially taking this compound that has a carbon-nitrogen double bond and making that compound with a carbon-sulfur double bond and with the multitude of amide structures in it, and this is more stable. Okay, so we initially, when we cleave it, we make this compound right here initially, probably just as the oxygen. And if you were to, under mild conditions, you can probably isolate something like this, but this is usually this is done under relatively harsh conditions, and you end up with this structure right here. Okay, all right, so Edman degradation is really great for looking at the N-terminal amino acid, but I'm going to just say that there are many more techniques that you would really need to employ to actually sequence a protein. Okay, and that is a discussion better left for a course in biochemistry. Okay. All right, so let's just predict what we would get here. Predict the protein and hydantoin products that you would get from the following reaction. Recognize that the N-terminal amino acid is tyrosine. Next one in the chain is an isoleucine, followed by a cysteine, followed by a tryptophan. Okay, all right, so let's do the Edman degradation. All right, so here we can see that our N-terminal amino acid is tyrosine, so we're going to clip off that whole unit, and we're going to see that this group right here is going to be the substituent on the hydantoin. Okay, since it's a tetrapeptide, it's going to end up being a tripeptide after we do the Edman degradation. So we're going to see two products. Okay, so we're going to see as our protein product isoleucine, cysteine, tryptophan, and as our hydantoin product, we're going to see that product right there, which has, again, the hydroxybenzyl unit of tyrosine incorporated into the hydantoin structure. Okay, and I think this is where we're going to end this video right here. In the next one, we're going to look at making peptides.